Active transport is different in that it's moving materials against their concentration gradient. We're trying to fight diffusion here. And because we're going against the natural energy of the environment, right? You're trying to go up the stairs, you're trying to go up the hill, you're trying to go against the flow of the river. You have to provide energy. So this does require energy on the part of the cell. And cells use ATP as the energy currency for this process. Active transport does require protein helpers in the cell membrane, so it is a carrier-mediated process. So it's like facilitated diffusion in that we're going to use a protein helper in the membrane. It's different from facilitated diffusion because substances are moving against their concentration gradient and we have to supply energy. So one example of active transport are ion pumps, and we see this in the picture on the left. Now, this is an example of a proton ion pump, so it's pumping hydrogen ions or protons, if you will. And if you notice in the picture, we're taking the hydrogen ions from inside the cell and moving them out, but there's already a lot of hydrogen ions outside the cell, right? So according to diffusion, the hydrogen ions want to come into the cell, but you don't want that to happen because that's going to change the pH inside your cell, right, and cause it to become acidic. Instead, you want to remove the hydrogen ions from the cell. And so we have to move the hydrogen ions against their concentration gradient. So this requires ATP energy and a proton helper in the cell membrane, and a protein helper in the cell membrane to facilitate the process along. So this is active transport. Another important active transporter example is the sodium-potassium exchange pump, also sometimes just called the sodium-potassium pump. The sodium-potassium exchange pump can move both sodium and potassium in different directions, but both are moving against their concentration gradients. So this does require ATP because we're moving these ions against their concentration gradient. The sodium-potassium exchange pump is made out of protein and that's embedded in the cell membrane. And this is another example of active transport. Sodium-potassium exchange pumps are really important. We find these in almost all cell membranes of all cells, but they're really important for your heart muscle and your skeletal muscles and your nervous system cells. Without the sodium-potassium exchange pump, none of those cells would work properly. And scientists estimate that more than 50% of all the calories that you consume actually go to make ATP to run just your sodium-potassium exchange pumps in your body. So that makes them really important. Bulk transport is very different than both passive transport and active transport. This is a process where the cell membrane is going to be wrapping around large amounts of material. And it's going to involve things called vesicles. Vesicles are little membrane-bound sacs that we find inside of cells. And they look like little bubbles in most of the pictures. When it comes to bulk transport, we have two types. The type that brings stuff into the cell, which we call endocytosis, and the type that brings stuff out of the cell, which we call exocytosis. When it comes to endocytosis, we have further subcategories. Phagocytosis is the cell eating, which means the cell is using bulk transport to bring in large particle matter into the cell. Pinocytosis is the equivalent of the cell drinking, right? Pino means to drink. In this case, the cell is bringing in a large amount of substance, but it's almost entirely liquid without any of those large particulate matters in there. The third type is called receptor-mediated endocytosis, and we will not discuss that in this class. But I just wanted to mention it in case you came across it in your reading. Let's take a look at some pictures. Right? So I have three panels here, okay? The top, the middle, and the bottom. One of these is showing exocytosis. The other two are examples of endocytosis, one being phagocytosis and the other being pinocytosis. Pause the video here and see if you can identify which is which. Remember to read the pictures from left to right. So in the top panel here, we see the process of phagocytosis occurring. The cell membrane is wrapping around a large particle matter, a bacterial cell, and bringing that into the cell. Right? And you can see we end up with a vesicle inside of the cell, and that vesicle contains the bacterium. The cell will then break down the bacterium, maybe using some of it for food, or maybe using some of it to start an immune system response. The middle panel shows exocytosis. Right? The cell has a vesicle filled with some sort of product that it needs to get outside of the cell. Once the vesicle fuses with the cell membrane, the contents of the vesicle are then released outside the cell. Exocytosis is how we release most of our neurotransmitters. So neurotransmitters are chemicals used by the nervous system, right? And they are released through the process of exocytosis. And the bottom panel is showing pinocytosis, where the cell membrane is wrapping around mostly liquids with some small solutes and bringing that into the cell, in the process producing a vesicle inside the cell containing the contents of the liquid.